Let's jump right into it. We're continuing with Lecture 3 on gametogenesis. The slide on oogenesis, the female gametogenesis. And we'll push on through this lecture, the remaining portion of Lecture 3, and jump into Lecture 4 as much as is possible. We've discussed mitosis, meiosis, and the male gametogenesis. We have a feel for the terminology, starting with the gonia cell, the reserve cell with the mitotic capabilities, and then proceeding to a primary and secondary developmental series and meiotic division series of the cells differentiating into the gametes. So obviously in the female we are going to focus on the ovary as the reproductive organ, the gonad, and see some of the histology of that and a variety of illustrations. The term oogonia refers to that reserve cell in the female gonad, and it's a primordial gonad, basically, that they will begin to emerge within and divide, and we'll study that later when we focus on the reproductive system. Stated before that the oogonia do not persist in the female gonad really much after birth or neonatal periods. So they are really embryonic and fetal cells, and what we discuss as far as primary oocytes on through really begins to occur at puberty, as we had stated before. So at birth there are many primary oocytes and no oogonia. So the oogonia are those diploid cells that undergo the mitotic division and lay the groundwork for plentiful primary oocytes at birth in humans. So the primary oocytes are the diploid cells. Remember, they have both homologs, but the homologs should be in the duplicated state. And they are the ones, of course, that will undergo the meiosis 1. And they're surrounded by supportive cells called follicle cells, and will develop a nomenclature for this population of cells that surround the follicle as the follicle itself undergoes development. So there's two separate stages or series of developments that are occurring. One is the meiosis and the cell division, the cytokinesis, to produce the gamete cells, the female gamete cells. And then the second is this population of cells that surrounds the primary follicle undergoing a series of developmental stages, so that we'll end up focusing on three different cell clusters, layers, populations, if you will, ultimately in our discussion of the development of the follicle. When we proceed to the discussion of the menstrual cycle, we will talk about follicular recruitment, follicular development, and then obviously ultimately the development in humans of the single follicle that will become the mature or Graafian follicle to undergo ovulation under the appropriate stimuli. So the recruitment of follicles is from this pool, this tremendous pool of primary oocytes. And also, therefore, surrounded by their cells, they are the primary follicles. So that'll develop into secondary follicle containing the oocyte through its development. So this oocyte is suspended in prophase 1. Primary oocyte completes all the meiosis 1 shortly before ovulation, and then at ovulation begins the meiosis 2 division to metaphase. So as the follicle develops, meiosis will proceed. The division of the cytoplasm will be unequal in the case of female gametogenesis, oogenesis. So we've all heard the term of the polar body. So the nuclei will be the same, whether they have just one of the homologs of the duplicated chromosomes or a single sister chromatid that's now become an unduplicated chromosome within the final oocytes, the ova. But the amount of cytoplasm will differ. The polar bodies that are generated are going to have just a thin rim of cytoplasm, whereas the cell that is ultimately going to become the mature ova, more capable of that, will have the bulk of the cytoplasm. Remember that upon fertilization, there's really no opportunity for the cell to create new cytoplasm and new energy stores and things like that. So everything has to be there in the cytoplasm of the ovum. So stored lipids, amino acids stored somehow, either as proteins that can be degraded to free up amino acids, or however it's stored, glucose stored, however it can be, all the molecular mechanisms to undergo that process, cytoskeleton for division, everything must be available, including any messenger RNA protected so that it won't be degraded if it is needed for any translation along the way, because essentially, officially, the genes will not be transcribed. So the secondary oocyte is the functional haploid cell, which will undergo meiosis II when fertilized, and the ovulated secondary oocyte is surrounded by the follicle cells and a zona pellucida. So there is a layer of complex carbohydrates laid down outside the oocytes as they're developing, and it becomes the zona pellucida. This is part of the barrier that we talked about is the task of the acrosome to dissolve. The cell, which has now completed both meiotic divisions, is a fertilized ovum, not necessarily fertilized, an ovulated ovum, plus 3 residual bodies. So we still end up with 1 primary oocyte and 4 gametes, haploid gametes in the end that contain the single representative of each homologue of the chromosomes, and these chromosomes are unduplicated. In this slide, we can take a look at the life of a human egg to remind ourselves when throughout the lifetime, the timeline of development, will we find certain types of cells, obviously focusing on the oogonium. So the primordial germ cells will emerge from kind of a surprising area in the yolk sac and will migrate through the undifferentiated connective tissue cells, the mesenchyme, within the early embryo, and they'll end up at a spot called the genital ridge that, of course, we'll study later. These oogonia will settle in there, so all the other cellular components of the ovaries will develop from the mesenchymal cells in that area and surround the oogonia and support them in their mitotic division during embryogenesis to produce the primary oocytes arrested at prophase 1. And these are the population available for recruitment throughout the life of the reproductive life of the individual. So at puberty, ovulation will begin, and from this pool of primary oocytes arrested in the prophase 1, the secondary subpopulation of those primary oocytes will be recruited into development, and they will undergo that development, becoming the secondary oocyte arrested at metaphase 2. So meiosis I will continue to the stage of metaphase 2. Then with ovulation, the final division will occur, and if fertilization occurs, then of course the 2 pronuclei will undergo the fusion ultimately, and the pronuclear fusion will bring about the stage of development called the zygote. So we begin to introduce now specific terminology. The gametes, upon union and successful fertilization, in other words, conception, are called gametes until that fertilization occurs and is successful. Subsequent to that, the term for this concepsis is the collective term for this developing organism, the embryonic and extraembryonic membranes, placenta and stuff, throughout the entire developmental period until birth. So that entire concepsis at this early stage, even if there isn't any division yet, is called the zygote. Up until the point that within the first week or so that implantation has initiated and there is further development, then there'll be some different terms for the zygote, but it's still basically a zygotic stage up until the point we have a three-layered embryonic disc, really, and that is when we begin to refer to it as an embryo. Then it's an embryo until the 8th week, and then we use the term fetus. So many different terms to describe these very specific stages of the development, and I do my best to utilize the appropriate terminology as we proceed. Illustrations are always inadequate at representing real life, if you will, real biology. And we often see slides like this that make it almost appear like primary follicles will migrate as they develop and then continue to change after ovulation. And of course, this isn't true. There's, I'll say it's random, but no one really knows yet, as far as I'm aware. There's a pool of these primary follicles that will be recruited, and they will begin to develop, and ultimately only one of them will reach the stage of maturity, and those that don't reach this mature stage will undergo a spontaneous death process. The collective term is atresia. They will just disappear, disintegrate on their own, but the biological process for that type of cell death is apoptosis, where it's a gene expression dependent suicide, if you will, in that case. It occurs a lot in the developing fetus, especially, and throughout adulthood in limited cells. After ovulation, the cells surrounding the oocyte will undergo dramatic biochemical changes, begin to store cholesterol ester. Cholesterol ester has a yellow color, so it's obviously the corpus luteum. So there are many different components to the oocyte. What's the term for this thick connective tissue layer surrounding the ovary? The tunica albuginea again. A white tunic, thick layer. But there's additional detail about that I'm just not going to go into now. We see the attachment of the ovary to the suspending ligament, the ovarian ligament, and this is delivering the neurovascular bundle to the ovary. Excuse me, it's the suspensory ligament of the ovary. So then we proceed to this slide that has an illustration on the left, and that is quite simplified and focusing only on the events of the oocyte, and on the right-hand side, the events of the follicle collectively. So we will proceed through this series. Before birth, the eugonia is the stem cell. So you see this term stem cell appearing very early on. Mitosis will produce the primary oocytes, and there will be growth and development, and these continue to be haploid. And ultimately, of course, as it proceeds through division, there will be haploid, but it will have the full component of both homologs, the homolog pairs, as duplicated chromosomes and undergo the meiotic division as we had described previously. And then from childhood, in childhood it's inactive, obviously, and then from puberty to menopause, there will be the selective recruitment of those that will go on to develop through the lineage, ultimately to produce the 4 haploid gamete cells, each of which has one set of one of the sets of the homologous, the homologs, as unduplicated chromosomes. So it's here at when meiosis 1 is completed and the primary oocytes each month, that the division produces the first polar body that is smaller, as we said, but the nucleus is essentially the same. And then the secondary oocyte as a product is arrested at the metaphase 2 at ovulation. When, of course, fertilization can occur, meiosis 2 will be completed. And here we note that clarification that the contact with the sperm is essential for that final division to produce the final polar body. And of course, the first polar body also divides into 2 polar bodies. So we start with 1 primary oocyte, end up with 4 gametes. The same process that we had seen before, just an unequal cytoplasmic division, producing the ovum with plentiful cytoplasm so that can be in the zygote stage distributed to the different cells when it divides into 2, and then 4, and then 8, and then 16, and then somewhere after 16 it'll start producing a little bit of new cytoplasm along the way. The other illustration is showing the follicle cell development. There is not much visually that occurs with the follicle cell development as it proceeds through this follicle maturation process, but biochemically there are many changes and some final or terminal cell differentiation, so that we have supporting cells surrounded by the female gonadal steroid producing cells. So the numbers of cells in that area ultimately at the later part of the primary oocyte development will increase, and these different layers will begin to undergo development. So a lot of mitotic activity in these cells and recruitment of new cells into that pool. And then one of the collection of recruited follicles will go on to develop into the mature, the vesicular or the Graafian follicle, and that will be the one to ovulate and produce the mature ovum that is capable of fertilization along with the 3 polar bodies, and some of those follicular cells that produce the corona radiata, as we had mentioned. Thus, we can look at a couple of histological stains of the human ovary. And on the left panel, we see several primary follicles, so you can visually see a single layer of follicular cells surrounding the ovum. So you see the cytoplasm here, this will be the primary oocyte in a primary follicle, and then this is the nucleus. And there are several other ones. They might be cut in different levels of section through the cell. So if there was a plane of section through over here, it might look smaller, such as this over here. So some of these still might be in different stages of being recruited as well, but you have to realize that you can get different diameters of these structures. If you picture a basket filled with basketballs, and if you just took a knife and cut through there, you're going to cut through the basketballs at different planes. One you might go straight through the center, the next one you might just catch the very tip of it, another one you might kind of go a little bit deeper. So very rarely are you going to have a nice cut straight through the midpoint to see the full diameter of the structure. This image on the right-hand side has a blue arrow and a green arrow, and it shows a primary follicle with an acellular layer that surrounds the oocyte. And that acellular layer is the zona pellucida. So the zona pellucida is beginning to be deposited in this area. The follicular cells are undergoing development. Can anyone tell me what this structure is? Pardon? It's a vein filled with red blood cells. So these are all little red blood cells in there, and then this is a significant proportion vein, a little bit larger than a venule, filled with red blood cells. Then the tunica interna is responsible, it surrounds the follicular cells, and it's responsible for secreting the steroids. So there's going to be a layer of cells, tunica, that forms. It's ultimately going to separate into two layers, and it contains, remember, we're just seeing it in two dimension here. This is a basketball surrounded by two additional layers of structures, some multicellular layer and another that's just beginning to develop at this time. This image is a secondary follicle. We know for certain that we can refer to it as this because we see within the follicle that the follicular cells are starting to separate, and there seems to be a space in there. This is a space that's going to be filled with the liquid, the liquor folliculi, and it's that fluid that is essential to build up the hydrostatic pressure at the time of ovulation to rupture through the tunica albuginea of the ovary. So we see that the follicular cells begin to position themselves at the periphery, separate to form this space, and some of them stay around the ovum, and of course the green arrow is pointing to the zona pellucida that we had seen before. So it begins to develop and it persists. Now at a slightly later stage, the tunica is beginning to divide as the cells differentiate on an outer layer and an inner layer. So there's a tunica interna closest to the follicular cells, and a tunica externa more peripherally located on the outermost edge of the secondary follicle. So you can see that the cells within the tunica interna are a little plumper. They don't seem to be kind of flattened out. They're not as rounded and plump as the follicular cells, but the tunica externa cells are slightly more flattened. You can see the elongated nuclei, and that the cytoplasm kind of is flattened out and stretched. So characteristically, they are distinctive. They play an integral role in generating intermediaries for the biosynthetic pathway of estrogen. So one layer is going to produce the intermediaries from the precursor up to a certain point, and then that'll diffuse away and become available for the next cell to pick up the enzymatic series to convert it ultimately into estrogen. And we'll look at those series a bit later. What is the precursor for the gonadal steroids? What made up the yellow stuff that I mentioned? Cholesterol. So the yellow part of the what's it called? The corpus luteum. Luteum means yellow. Corpus means body. The yellow part of the corpus luteum that these cells all differentiate into after ovulation is made up of cholesterol ester. So obviously, after ovulation, that one remaining follicle is just going to have this tremendous synthetic power to sustain the synthesis of the gonadal steroids. At that stage, of course, more progesterone than estrogen. Remember that up to the point that we get the mature follicle, we have a recruited subpopulation of primary follicles that have some follicular development and increasing capabilities of producing the estrogen and some progesterone. So collectively, they will contribute to the plasma levels of estrogen and progesterone throughout the menstrual cycle. But after ovulation, there's only one follicle left, actually the corpus luteum, that is responsible for generating all the progesterone and estrogen essential for sustaining the uterus, or to promote gestation, to sustain the endometrial lining of the uterus and the pregnancy up until the point that the placenta begins to take over. So here's that oogenesis, the sequence of events. The oogonia undergo mitosis, forming more and more cells that continue to divide until there's a differentiation of all of them to the stage of the primary oocyte, frozen or suspended in prophase 1. Meiosis 1 brings it to the secondary oocyte with the first polar body. Second meiosis is at fertilization. I'm correcting myself there. And the fertilized mature ovum is present, and then the second polar body is there. The first polar body typically will undergo its second division to produce the 4 gametes. I almost said identical, but they're not really identical. We know that. Now we proceed to a histological slide that is at quite a low magnification. I draw your attention to the lower right-hand side. There is a scale bar here of 1 millimeter. So you can see that the mature Graafian follicle approaches the dimension of 1 millimeter. We don't know if this is cut right down the center of it, but I would bet that from this border to this border, it's approximately 1 millimeter. And these are human, obviously, post-mortem, or at least maybe excised or surgically removed. What is the term for surgical removal of the female gonads? Oophorectomy. This is the ovary. Actually, it isn't. Hysterectomy is just the uterus, but it's typically used for so a complete hysterectomy is a hysterectomy plus an oophorectomy. So the OOPH, the oopho, is referring to the ovary. So oogenesis, or oogenesis, however you want to pronounce it, refers to the development of the ova. And I apologize if I didn't phrase that question appropriately. Yeah, I said the surgical removal of female gonads, right? So the uterus is not a gonad. So here we see this diverse distribution and population of follicles at different stages. Some are still primary follicles, maybe not recruited. Others are larger and undergoing development. So these larger follicles here are probably primary follicles that were recruited. They're contributing to estrogen and progesterone, but we already have a mature follicle that has been selected, so they are going to begin to undergo this apoptosis, the atresia, atresia, and begin to disappear. So ultimately they will become atretic or atretic follicles, and they are no longer available for recruitment and development and have been permanently removed from the pool of primary follicles. So the one mature Graafian follicle, and again, this is human embryology. We know there are some species that are capable of multiple ovulations. Occasionally in humans there are multiple ovulations, but not as many unless you're being boosted with the appropriate drugs for in vitro fertilization. So obviously the boxed areas are some of the areas we're going to zoom in and take a look at. First, let's look at the edge of a mature follicle. So there is the antrum, the term for that space within the follicle. So the follicle, or follicular antrum, is that space, the cavity, that is filled with fluid, the liquor folliculae. And the periphery of that antrum is surrounded by a multicellular layer of these follicular cells. Now we're at a point of development and differentiation where we call them granulosa cells. So the granulosa cells are really those follicular cells that we referred to originally. And that term can be used generically back to the initial developmental stages. Cells like this, this really isn't an epithelium, but it represents a similar type of a cell layer, will have a basement membrane. And then outside that basement membrane is the theca. So there's a theca interna and a theca externa. Then the tunic. And on the outer surface of the ovary, outside its tunica albuginea, is a layer of simple epithelium, simple cuboidal epithelium, simple meaning one cell layer thick. This is multiple cell layer thick. This is single cell layer thick, so it's simple epithelium. And they're as tall as they are wide. They're cube shaped, at least in the two-dimensional sense. And therefore a simple cuboidal epithelium. It's actually called the germinal epithelium. That was originally thought to be the source of the ova. I don't know what they thought all these other things in the ovary were, but I'm sure it confused them. So the theca will be responsible for the gonadal steroid production in a cooperative fashion. Now you noticed in this slide, and even an earlier one when the antrum was just beginning to develop, that some of the granulosa cells, follicular cells, what we called them then, stay around the periphery of the zona pellucida, the developing ovum, and they will remain there, but the ovum will not lose its attachment to the peripherally displaced granulosa cells, at the periphery of the antrum filled with fluid. There will be a column of these cells remaining to suspend the ovum and the surrounding granulosa cells within that liquor folliculae, the antral compartment. And the term for that column of cells is cumulus oorophoros. So we see the oopho coming in here to refer to the ovum, which of course is in the ovary. Thus, and this is this correct? Terminology on there? I didn't label the slide. I kind of borrowed it from the internet, but now that should be a secondary oocyte by this time at least, right? If it's proceeding toward ovulation, it should be proceeding toward the metaphase 2 to be suspended there for ovulation. So up above it is labeled correctly as a secondary oocyte, should be frozen in the metaphase 2, and this is after ovulation. So we see that a portion of the granulosa cells surrounding the secondary oocyte and the mature or Graafian follicle will accompany the secondary oocyte out into the peritoneal cavity and then ultimately into the fallopian tube, of course. And this is called the corona radiata. So these are the cells, the granulosa cells that accompany the ovulated ovum and are responsible for interacting with the sperm when they arrive. The sperm have to be able to make contact with them, stick to them, and work their way through, break the tight junctions between those corona radiata cells, and work their way down to the zona pellucida and proceed from there. And the acrosomal enzymes, of course, are essential to dissolve a hole through the zona pellucida when the membrane of the ovum is contacted. There's a change that we'll talk about later, and this triggers the final division of the secondary oocyte. So this orangish red colored highlighted area is the cumulus oorophoros. That's the zona pellucida, and then the primary oocyte is here. These series of events at conception are illustrated in this slide. We see the corona radiata, so an irregular border of these granulosa cells that accompanied the secondary oocyte. It's suspended in metaphase 2, The first polar body should be present inside the zona pellucida. So the zona pellucida is roughly the eggshell, but it's obviously not the calcified type of an eggshell that we see with the chickens. But it's essentially the same as that. Although fertilization is should already have occurred in that case. So the corona radiata cells are the first barrier for the sperm. There's now some evidence that the ovulated ovum with the corona radiata secrete chemicals that are attractants to the sperm. There is some selectivity, especially at the zona pellucida and at the membrane of the ovum. But once contacted by one or more, the ovum will obviously be triggered into the second meiotic division, to complete that. And so there's now two polar bodies. The second polar body is here. The first may or may not complete its division. And one sperm will be allowed to fertilize, and the others will be rejected in essence. And the male pronucleus will enter, obviously, and you have the female and the male pronucleus, the first and second polar bodies, whether or not the first divides. And then here in panel D, we see that the fusion of the pronuclei occur, and there would be the junction of the homologous pairs of the unduplicated chromosomes from each corresponding gamete, the paternal and maternal, and mitotic division will begin to occur. So at this point, they're kind of showing this sperm inside the ovum. So at fertilization, this is now the zygote. So we're seeing the zygotic events after fertilization up to the point where the first mitotic division of the zygote will begin to occur. Then here we have a slide with a couple of questions on it. You can see Q1 and Q2. So Q1, what is this structure? The primary oocyte, right? Because we don't have an antrum. It's just starting to loosen up a little bit there maybe in the follicular cells or granulosa cells. But it'd be primary. So this is a primary oocyte. There's its nucleus. This is the cytoplasm. And this is the zona pellucida. And then Q2 is the granulosa cells, follicular cells, either would be accurate. Looks like some of the theca is beginning to approach cells that will produce the theca are in the appropriate location. Here's another slide, and we are asked the question about the highlighted structures. So obviously it's this dark pink that we're referring to. And what would that be? What would this layer be? I know that's not the question, but what would this layer be? Let's figure out what it is before we try to answer the question. The zona pellucida. So that's the zona pellucida. And the question is, it appears between what two layers? Or what two structures, right? So the oocyte and the granulosa layer. Not to answer the questions for you, but anyway, that's what you're supposed to be doing on your own as you discover these questions embedded within the lecture. So this one you should be able to answer immediately. The highlighted cells. So even though this looks like it might be highlighted, it's not cellular. It's an acellular layer, cell-free layer. This is the highlighted. This is the blue stain of the histological procedure. So that's what all the cells would look like if it wasn't digitally altered here to be red or orangish. So from what cells do they originate? Again, the same as we saw in the previous one, the granulosa cells. They are actually granulosa cells. Then here's an illustration. Obviously, this is primed for posting on the online testing. So 1, 2, 3, 4, 5, 4, 5, 6, 7, 8, 9, and basically these follow in that sequence. I won't follow them in sequence, but I'll orient from the oocyte on out. So this is the oocyte, the zona pellucida. Then the granulosa cells will be everywhere else. But there's some additional terminology that we see here. This number 9 is the spacium perivitellinum. Obviously, that's nothing that we're going to focus on, but it is a space around the vitellinum, basically the surface of the zona pellucida. So that's one that we haven't seen, but everything else we've seen, the granulosa cells here that will contribute to the corona radiata. So this part here, just on the opposite side, would be the origin of the cumulus oophorus, of course. So we've taken care of everything in here. Then these are the granulosa cells. So 1, 2, 3, 4, 5, 6, membrana granulosa. Then obviously 1 and 2 are going to be the theca externa and the theca interna. So 4 comes all the way down here. So at this polar end of the ovum, where the granulosa cells are still all connected together, this is the portion that will ultimately become the cumulus oophorus. Well, we have about 7 minutes or so. I'll just get started on the lecture 4. In lecture 4, we are going to discuss the female reproductive system and the menstrual cycle. Obviously, we are not going into the most extensive detail to come up with this. Well, it's still going to be an overwhelming list of nomenclature that you and terminology that you have to learn, but we're going to limit it some. So there isn't quite so much of the gross anatomy. Then we'll proceed to a discussion of the menstrual cycle. So the secondary organs in the female reproductive system are the uterus and the cervix. Obviously, the primary ones are the gonads, the ovary. The uterus is going to have a body and a cervix. The body is going to have a perimetrium, peri obviously around. The metrium is referring to the layers of the uterus itself. The myometrium we know is the middle layer, the smooth muscle layer. And then the endometrium, endo meaning inner or inside, is the inner layer. Thus we proceed to this illustration of the internal female reproductive organs. The primary organs, the gonads, secondary organs that we had mentioned, the uterus. We know the fallopian tubes are attached to the uterus. And here's that suspensory ligament of the ovary that I had mentioned to correct myself, because that is indeed not the ovarian ligament. This is the ovarian ligament, so I needed to correct myself. It's a ligament that suspends the ovary in the pelvis and carries the neurovascular bundle. So we are oriented to the ovary there. Obviously, at ovulation, the ovulated ovum is going to be close to the finger-like projections of the fornix, the fimbria, and then it will be captured and proceed through the fallopian tubes toward the uterus. And we see the body of the uterus here and the cervix here. The cervical canal from the external cervical os opens into the internal cervical os, and then the uterine cavity is present, contained within the body, and then this portion of the uterus that projects up above the connections with the fallopian tubes is called the fundus. So there are different portions, and our objective at the moment is to proceed to a histological examination, a micro anatomical evaluation of the layers of the uterus. Starting on the outer layer, we see it is covered with a thin layer that has a layer of simple squamous epithelium on it, called the parametrium. And so these are simple squamous cells, look like a fried egg, a sunny side up fried egg, and they're just all attached to each other and form a continuous layer over the uterus, as it does all the structures within the abdominal pelvic cavity. It's basically the same as the peritoneum, except on the uterus it's referred to as the parametrium. And the vasculature penetrates through the parametrium. Then the next layer, there's a little layer of connective tissue holding those simple squamous cells in place. The next layer is the myometrium. What pituitary hormone targets receptors on the myometrial cells, smooth muscle cells? What's Pitocin? Pitocin is the term for the drug that's used to induce labor, but what's the name of the hormone that comes from the pituitary that does it naturally? Oxytocin. Okay. So oxytocin's coming through here all the way from the pituitary somehow. Pituitary to the heart, through the lungs, back to the heart, out through the heart, down through the abdominal aorta, all the branches until ultimately the uterine artery delivers it in through the myometrium, the capillary beds, diffuses out, bounces around until it bumps into its receptor on the smooth muscle cells and triggers and sustains the rhythmical contraction of the myometrium. These arterial supplies continue down to arteriolar level, penetrating the connective tissue layer, separating the myometrium from the endometrium. The endometrium is multicellular. We'll describe all the cells as we proceed. I'm not going to try to do it all at once here today, but we see that these arterioles branch and form these spiral arteries or arterioles that proceed all the way to the subjacent component of the endometrium, just subjacent to the surface epithelial cells of the endometrium that line the uterine cavity. We see also that there are glands in the endometrium with their ducts opening into or onto the surface of the endometrial layer, and obviously they will ultimately secrete glandular secretions, a nutritive mucus, seromucus secretion onto the surface, and this contributes to the stickiness for the ovum, fertilized ovum, zygote, whichever it would be, is going to make contact when it enters the uterine cavity and ultimately stick with this, and then this is the supply for diffusion of the early nutrients for the developing zygote. So this glandular secretion is important. What hormone triggers the secretion of these glands and promotes gestation? Progesterone. It also, of course, is going to contribute to the sustenance of the endometrial layer. If you block progesterone receptors, what happens to these cells in this layer? They will slough off. It actually causes a vascular event that we'll describe in the next lecture. Causes the shedding of the menstrual, the functional layer of the endometrium during menstruation. So we'll start with this slide and get into it a bit more detail as we proceed through the description of the female reproductive system, focusing predominantly on the uterus, and then we'll go into the female hypothalamic pituitary gonadal system with the menstrual cycle.
about the metaphase 2, how it stays suspended until it awaits for the homologous chromosome to sperm, correct?
No. Metaphase 2 is during meiosis 2.
Yeah.
And at that point, the sister chromatids stuck together of just one pair of the homologs is at the equatorial plane of the second